Thank you, Cora. It's very nice to have you introduce me. I think I'm sure I'm not alone among professors, but I would say that the top part of the job, the most rewarding part of the job, is to work with wonderful graduate students like Cora was. I'm also very happy to be here to honor Carl. Carl was very important to me personally, and of course his work was really important in my professional development. And it's an honor to be able to be here for this kind of meeting. My talk is a variation on a talk I gave 2 years ago, which was at the ENAR meeting of the Biometric Society. And that too was a presidential invited lecture, and that was a big honor. It brought back to me sort of my youth, and in fact, having Cora introduce me sort of brings back my youth too, because it was such a long time ago when I was teaching Cora. But what I really remember about the Biometric Society and the ENAR meetings when I was younger is that I would go with many of my contemporaries and talk to them, and one of the things that we would eagerly look forward to was hearing the keynote address of one of the stalwart senior statisticians, and really never in my wildest imagination could I have visualized myself someday being that old. But here I am, and I think I do pretty well, though I don't know if you know this, I was recently diagnosed with CRS. I can't remember shit. One of the many unfortunate problems of getting older. What I want to talk to you about today is the brain sciences, neurophysiology, which seeks to understand how neural activity produces behavior. Billions of individual neurons, collectively, somehow, receive our sensory information, our sight, hearing, touch, smell, taste, and integrate it, and use it to create feelings, memories, thoughts, decisions, and actions. While this complexity remains staggering and imponderable, I find the subject fascinating in part because there's been immense progress in understanding the nervous system over the past 100 years, with neuroscientists attacking problems at multiple levels, from behavior down to systems. Let me just point to this. Behavior is up on the left here. Behavior to systems, and then around to neurons, ion channels, and then all the way around to genes, really using tools from diverse disciplines. I'm going to talk about the level here of circuits and individual neurons. And I'll start with a little bit of background on neural coding and spike train modeling. I'll explain what those are. A quick overview of some recent things I've worked on involving neural synchrony, the phenomenon of two or more neurons firing at roughly the same time. And then as I get toward the end, I will quickly make a comment about visualization in general and in diffusion imaging in particular. And I will use all of this as an excuse to talk about what is statistical thinking. So the general problem of cognitive neuroscience is to discover how neural activity produces behavior. The more specific problem of neural coding is to elucidate the representation and transmission of information of the nervous system. This is a quote I took from a nice review from 1968, and these words are chosen to connote a technical kind of approach to the subject. Representation, transformation, information, they all have technical aspects. So to say more about this, I have to give you some background, and I always like to start at the beginning and make sure everybody's kind of on the same page with this. So now I think most of you already know this, but the brain is located in the head. And you also will remember that neurons communicate with rapid electrical discharges, voltage jumps here, which take place over the course of about 1 millisecond, which on a behavioral time scale of several hundred milliseconds essentially become a set of points in time. These are called spikes, and this is called a spike train. So this is the thing that I've spent a lot of time dealing with, the kind of data I've spent an awful lot of time dealing with. So the one natural way from a mathematical and statistical point of view to deal with data like this is to represent them as point processes. Again, it's just an erratic sequence of points in time. But the way the data actually comes is a sequence of zeros and ones, 1 whenever you have a spike, 0 when you don't, in some time-binned version, and this is how we actually analyze the data as a binary time series. You can go back and forth between continuous time, point processes, and discrete time, binary time series, and of course the two probability distributions you would get in both continuous time and discrete time, they have to be consistent as they are. This comes from the probability of a spike in a particular time bin is approximately this point process intensity times the length of the time bin. That X there is a stand-in for covariates, and I'll talk more about that. So the point is you can do a discrete likelihood based on binary data or a point process likelihood in continuous time. It doesn't really make any difference. You get the same statistical analyses in the end. And we use point process likelihoods to get formulas, essentially, that might simplify things, but otherwise, the vast majority of what we do is in discrete time. And the tool is just modern regression, by which I mean you start with some function of variables to get y, and then there's noise involved in some way. And here it's point process regression, which is really just, if you're not familiar with it, it's just, think of it as logistic regression for binary data, or what I prefer to call it Poisson regression. The probabilities in any time bin are very small. So 0.01 would be a typical value. It doesn't matter whether you do Poisson or binomial based. They're essentially, for practical purposes, the same thing. And we use all of the GLM technology that was built up over the past, whatever it is, 20-some years, or really 30 years now, to do this analysis. And it's really very familiar. So let me go back to this question of how neurons represent and transmit information. And the first answer, historically, is by modulating firing rate. And there are several kind of landmark sets of work identified here with years and people. These people all get the Nobel Prize for working in different systems on this subject. And I'm going to show you actual data from the Hartline case, the Hartline paper that was cited there. He worked on the horseshoe crab, which some of you may know from if you vacation in New England, for example. You see these things and they're really cool animals. As a little boy, I loved them because they're about this big with the tail, and they had armor on their back and a weapon behind them, and they're fun to play with. But they were of interest for neurophysiology because their eye is very accessible, and even more importantly, the optic nerve is accessible and it's easy to record from. So this guy Hartline would shine a light on the retina and then record from the optic nerve, and he'd get one neuron. And here are shown some of the data from his paper where when the light is bright, you see this neuron firing very rapidly, and as the light becomes dimmer and then very dim, it will fire less frequently. So again, this is spike trains from one neuron. The point here is that the rate at which the neuron fires is what's coding the stimulus, carrying the stimulus information, the strength of the stimulus. It could have been that the waveform changed, but it doesn't. This is called the receptive field for that particular neuron, the place you have to shine the light on the retina to make that neuron fire a lot. That's called the receptive field. I'm going to talk about it again in a second. So when you move to primates, here's a cartoon picture, but it's showing the way the experiments are done in primates and non-human primates. Again, electrode goes into the brain, in this case the primary visual cortex, which is in the back of the head here. One neuron being recorded. This shows the monkey being alert. Sometimes they're anesthetized, but a lot of times they're alert. They don't feel this. There's a light that's now shined on the screen in front of them, and the place where you have to shine it in order to make the neuron fire a lot is called the receptive field. Now I'm going to show you actual recording from 1963 or so from the Hubel and Wiesel lab at Harvard, where they move a light, just like in this cartoon, they move a wand of light across the screen, and you will hear the neuron firing when the light goes through the receptive field. And they're just trying to find out where is that receptive field. They're going to draw it on the screen. There you have it. There's the receptive field for that particular neuron, and I like to show this because, first of all, it's illustrating that firing rates carry a lot of information, but also you don't always need statistics, right? This is just a very straightforward visualization device and gives as much of the information as they wanted to show. However, what is firing rate? Well, the naive neurophysiological answer is it's just the number of spikes divided by the time over which you record them. But, and this is used in a very important and commonly employed device called a visual display called the peristimulus time histogram. Here, the first line is one spike train. The second line is another spike train from a repetition of that experiment. It's as much as possible exactly the same experiment. This is 15 different replications or trials. 0 is a time when a visual cue came on the screen, and you can see by essentially averaging across or really counting up the number of spikes across any given time bin and displaying that as a series of bars here, normalizing in terms of firing rate per spikes per second, you get a sense of the time trend in terms of when the stimulus came on. So a very valuable display. And it is showing that number of spikes over bin size, firing rate. But that's, as you might immediately see, this is going to depend on the actual interval, the delta t. If in particular think about an interval that only has one spike, it's going to then the denominator you pick will entirely drive the answer of the, to what is the firing rate. So that's not good. Furthermore, as I'll explain in a second, it effectively assumes Poisson firing, and it could be highly variable. The bins are jumping all around. So I mean, we as statisticians want to smooth this, and that's really what we end up doing. The statistical signal processing answer is to say, well, it's really the instantaneous intensity of this process that you want to be calling the firing rate. We'll assume it's smooth, and so we introduce the point process framework, and now this lambda is what's the firing rate. So what we do is we build a model for lambda, for the intensity. Then we fit the model, usually with maximum likelihood. Sometimes, occasionally, we have to go to Bayesian methods. And we do statistical inference based on familiar stuff like the bootstrap. Again, modern generalized linear model technology is at the heart of it. So now to go back to this lambda function, let me elaborate a little bit. These covariates I talked about could involve a couple of things. One is one important source of X's here is spiking history, which is where the time series structure comes in, meaning neurons that fire at one point in time may behave differently because they just fired just after that time. So the probability of firing at a given point in time will typically depend on whether the neuron fired recently or not. This has to do with the actual mechanism of neural firing, the action potentials, and the ion channels that control that. And there's actually effectively memory because the ions don't clear out uniformly. There's a kind of a layover. And you see these so-called history effects, which are just binary time series covariates. So just how far back in time are we looking is what's going to go into this H for history. U is going to be other covariates. So the simplest case is Poisson, in which case the intensity function does not depend on history, and you have just independence. So here we have this equation I showed you before. We just take the easiest thing is just take the log of lambda to be some smooth thing like a spline, and then you smooth this PSTH like I showed earlier. And there's lots of problems where that works just fine, but there's also, and then I wanted to mention, we've also gone to more elaborate versions of the same thing where we build some complicated way of doing spline fitting, but actually garden variety spline fitting is just fine, as are other methods of smoothing. But then you get to the question of this non-Poisson behavior, where you have to sometimes, for certain purposes, take account of the fact that the history matters, the time series structure matters, they're not independent firing. And so here, if S is these S's indicate the time at which you have spikes prior to time t, first spike, second spike, and so forth, up to S lower star, which is the time of the last spike prior to t. If you want to build a model depending on that, it's actually going to be, in principle, very complicated because there's so many different possibilities for this previous spiking. And so what you got to do is build in some simplifying assumption, and the simplest thing you can do is say, well, suppose it's Markov. So now the firing rate would depend not only on time, but also the time since the last spike. An even simpler case is if those two effects are separable. So now you have stimulus effects in this first term here, and you have time since last spike or spike history memory effects in here, and the easy thing you can do is now just assume that each of them evolves smoothly and you model it with a spline, the point being that now you just throw it into a generalized linear model. Straightforward. When we first wrote about this, we introduced a terminology called Imi Model, which I won't explain. I'm not real happy with that name anymore, but I wanted to mention it because it's on this graph. So here's a probability plot, PP plot, where it's just a model, check on the model fit, and the point here is that when you use a Poisson, by the way, this is for one neuron, which is actually an olfactory neuron sensing smell in response to an odor, and if you do a Poisson process model here, allowing the rate to change over time but not allowing for any memory, you get strong lack of fit. And if you use a Markov model, there's still a little bit of lack of fit, but it's mostly sopping up this kind of memory effect. This is fairly typical of what we see. So over the time that I've been observing this literature, starting in about 1998, there's been a huge shift in the field. It's now much more sophisticated using modern generalized linear model and generalized regression technology. This allowed us to write a couple of reviews. Myself and my colleague, close colleague and friend Emery Brown at Harvard and MIT, we wrote a couple of these reviews in the 2004, 2005, where the main points were, look, you can use modern regression and time series method effectively in these kinds of problems, and so people already knew how to do Poisson-based analyses, and we were just saying, look, it's almost as easy to do the non-Poisson version. You just add on covariates that deal with the memory. So that's what I was saying before. Now there is so that's kind of the transferring fairly standard statistics into neurophysiology. But then there is an actual challenge even to sophisticated statisticians, which is to describe the functional connectivity among lots of simultaneously recorded neurons. So there are many different electrode arrays that people have devised, engineers have devised to do neural recording in animals. This is a very popular one. It's called a Utah array. It's relatively easy to use. It's relatively stable. It's got some disadvantages, but it's used a lot for certain kinds of recording. You can see it compared to a US penny. It's got 96 electrodes, 10 by 10, except the corners are taken out because that's where the wires go to hold it in. And here are the recordings that come off of all those electrodes. They're filtered either in low pass to get a particular thing that I'm not going to talk about, the local field potential. But for high pass filtering, you get these spikes. And here, it's a clustering problem. Each electrode picks up multiple neurons, and you got to figure out which. Remember, I said the waveforms don't change. So any one neuron on the effects of one neuron as recorded on one electrode will be repeated. So you now try to cluster the waveforms, and the ones that cluster together are the ones that come from a particular neuron. So here's an electrode that had two neurons pretty reliably recorded, and you can see two clusters, and once they're identified, then you go back and you find out what times those two neurons fired, and those become the spike trains. So there was somebody recently, a couple of guys I know recently published a paper in Nature Neuroscience pointing out that if you look at how papers doing simultaneous recording have evolved over time, the number of neurons people are recording has gone up exponentially. So that's what's on this axis on a log scale. So here, in the early 60s, they were recording one or two neurons, and now it's getting into the hundreds of neurons. And this just keeps growing, and their point was we're going to need a lot more statistical work to keep track of all this data. Here's an example of a data set that a student I was working with recorded. I was on his thesis committee because he had a lot of statistical stuff. He's actually in computer science, but he recorded the data in a monkey, along with another guy who was a postdoc at the time, who was a neurophysiologist. What I'm going to show you is a movie that's being shown to the monkey. The monkey's actually anesthetized, and these circles are receptive fields for individual neurons. There's over 100 neurons here. And when it's red, that neuron's firing. When it's black, it's not firing. And down below, you'll see the spike trains. And the point here is really just to show you what the data look like. This is a movie called Run, Lola, Run that was very popular back then. and you've got this rich binary time series, multivariate binary time series. The point would be, in principle, to find out what are all these neurons responding to and how are they interacting. And it's a much more complicated problem than those relatively simple problems I was showing you earlier. So what Ryan Kelly did is, he was the student I was working with and who recorded the data. He ended up building a sophisticated model. I'm not going to go into the details, but it's the log of the intensity or firing rate. There's some stimulus effects that are important, which characterize which visual stimuli seem to drive the neurons, some coupling effects across neurons, some history effects involving each neuron's memory, and then some global network effects which are effects that seem to drive lots and lots of neurons together. And altogether, there's maybe, depending on how he did it, there's anywhere from many hundreds to many thousands of parameters in this model, and that required a regularization effort, but it's fairly standard L1 regularized Poisson regression, and this we ended up publishing in NIPS. But around the same time, a couple other people did similar things. A really nice paper in Nature appeared using a similar technology, and also Nature Neuroscience. My point here is that we have tools now that are being developed in statistics to help with this dimensionality problem. Although I don't think it really solves all the problems we have with regard to inference, it does at least get us a step in the right direction. Now, I wanted to focus on the problem that I've worked on with this same guy, Ryan Kelly, over the past few years. He's now gone. He was a postdoc. He stayed as a postdoc with me for about a year and a half and did this work. Unfortunately, from my point of view, didn't go into the academic world. He had a two-body problem that forced him to go to Google instead. He was he's like the best programmer I've ever worked with. The point here is we have the phenomenon is two or more neurons might fire at almost the same time in a particular time bin here. Delta is going to be 5 milliseconds for us, 0.005 seconds. And here you see the spike train for one neuron, spike train for the other neuron, and there's a time bin where they happen to fire at the same time. Why would anybody care? Well, here's a nice display that's supposed to illustrate this. Look at this, I guess you'd call it visual illusion picture here. You can see this two different ways. You can see it either as a face behind this candlestick, or you can see it as one face looking to the left, one face looking the other way, and the candlestick's in between the two faces. So as you look at this, you'll see it both different ways. And the question is, what's going on in your brain as you flip between these two different perceptions of the same visual input, two different perceptions? So none of this is remotely proven, but here's a theory, and it's a very popular theory, of how the brain solves what's called the binding problem. Suppose you have 4 receptive fields here, 1, 2, 3, 4. The theory says, well, in this picture where it's candlestick and face behind, this one, number 2 and 4, are bound together. This one, 1 and 3, are bound together, whereas in the other case, it's 2 and 3 that are bound together, and 1 and 4, did I say that right? Sorry, 1 and 3 are bound together, and 2 and 4 are bound together. So what does it mean to be bound together? Well, maybe they have synchronous spikes. So over here, 1 and 2 have some synchronous spikes, and 3 and 4 have some synchronous spikes, whereas over here, 1 and 3 have synchronous spikes, and 2 and 4 have synchronous spikes. So synchrony may solve this binding problem, and there are people who seem to think that it will. This is often associated with oscillatory activity. This is part of the mechanism where there's actually an oscillation in the voltage which forces, when it's at the high point, neurons to fire. When at the low point, they don't fire, and this would tend to force synchrony across many neurons. To show you what I mean, this is from Emery's lab. Emery is both a statistician and an anesthesiologist, and he's doing a lot of stuff on the physiology of anesthesia. This is just one of the recordings he's made, an EEG on a patient who is undergoing anesthesia. They start off awake, and then they're induced, and they undergo surgery, and they recover. Here, in each case, you've got a time axis, and you've got a frequency axis. These are spectrograms. So this is different frequencies of the oscillation in the signal. The actual signal is shown up above. And the only reason I'm showing you this is to show that there's something quite different about awake and deep anesthesia, especially in these two bands. You have frequency activity around 10 hertz. You also have frequency activity in the very low, very slow waves. And you can kind of see that in this figure. You can see both of those things. That's the kind of thing that supposedly is going to carry these oscillations throughout different parts of the brain and could be contributing to synchrony. So now I'm going to show you some data from this anesthetized animal, and you're going to see some slow waves here actually in the data. These slow oscillations. This, again, it's anesthetized, so it's just like what's known about in humans. Here's a plot of, like the one I showed you earlier, a bunch of different trials for one neuron, and down here is this histogram shown. It looks, it's a sort of a slightly smoothed histogram. And then here's another neuron, same set of stimuli for a different neuron, and you see this response averaged across the trials here. And you can see dark bands, which have to do with the fact that the stimulus in this case is these gratings of light that I showed you drives neurons in the visual cortex, and it's moving at different angles, and that turns out to be important. For certain angles, certain neurons will fire more than for other angles. So they're responding to the stimulus. That's what these bands are. On the other hand, he made a different display, which is, it sort of superficially looks similar, but it's actually different. This is one trial of data for all the neurons. So each line is a different neuron. This is the second trial for all the neurons, the third trial for all the neurons. And what you see here is you see dark bands, but you see that they vary where they occur for different trials. In other words, these bands represent times at which lots of neurons were firing, but it's not the same on every trial, which means it's not locked to the stimulus. It's erratic. It's not stimulus connected. Well, these are the slow waves of oscillation that we see under anesthesia. So now if you're going to go back and try to understand synchrony and ask if it carries lots of information, obviously you're going to have a bunch of synchrony during these dark bands. Neurons are going to fire at the same time. That could just be caused by the anesthesia. So his whole thesis had to do with trying to look at synchrony when you get rid of this anesthesia effect. And just as an illustration, these are two pairs of neurons that actually are very different with regard to synchrony. I've shown the two neurons with the circles where there actually are synchronous spikes within 5 milliseconds of each other. And they look kind of erratic. In both cases, there's like a couple thousand spikes here, but only 30 or 40 are synchronous. And it turns out, even though you can't see it at all with your naked eye, that these pairs are very different. I'm going to explain that. So how would you analyze synchrony? Well, one obvious thing to try to do is to look at this as a bunch of 2x2 tables. There's 4 combinations. One fires, the other doesn't. The second fires, the first doesn't. Neither fires, or both fire. So now you could just aggregate across time and get a bunch of 2x2 table of counts. For n neurons, you get 2 to the n table of counts. So several different authors propose using log linear models to analyze this kind of synchrony. However, what I just said was you'd be aggregating across time. Well, if you aggregate across time, you're assuming stationarity, and you're assuming Poisson firing. There's no memory effects. Stationarity isn't going to hold in these cases where there's a stimulus that suddenly changes everything. And as I already said, we don't really think things are Poisson. So we wanted to develop a method that's like log linear models, but non-stationary and allows history or other covariates. And we also wanted this discrete time approximation to be essentially the same as what would happen in continuous time. And yet for that last point, we faced this mathematical impediment that I have to tell you, I don't mind telling you, held me up for like 6 years. I could not figure out how to deal with this, and we didn't work on this for a long time because I couldn't get over this stupid math problem, which was the problem is that the regularity conditions for point processes exclude simultaneous events. It's just one of the basic conditions. In order to write down all the machinery, you have to make that assumption. So the question is, how do you deal with simultaneous events when they're not allowed to happen? And so what I finally realized, it seems really obvious now, we had to just build a slightly different framework. I'm not going to go into it, but finally we got over it. And I'm pretty happy with the result. It's in these papers. The idea is really simple. If we want to know the probability of joint spiking, 2 neurons firing in the same time bin, suppose first that they're independent. If they're independent, then the probability they both fire is just the product of the probability that they each fire. If they're not independent, let's introduce another parameter that will measure the excess above independence in firing, in joint firing. If zeta, as I've labeled it here, is 1, we get independence. If it's bigger than 1, we get enhanced synchrony. If it's less than 1, we get suppressed synchrony. And there's some details about how we write down the probability model, but like I said, the concept is really simple. We write down probability of joint firing in terms of individual neuron firing, and then we have this like excess term over here, take logs, look at the continuous time version, and we have again a log linear model, which is in terms of we write down the stimulus effects for the first neuron, the history or memory effects for the first neuron, whatever covariates we have for that first neuron, same thing for the second neuron, and we always have left over this excess term, which would really be measuring the synchrony after we take account of all these history and covariate effects. So we're assessing the extent to which the covariates explain the synchrony, and to go back to that slow wave thing, if we want to get rid of the effects of slow waves, then the idea is you introduce the slow wave as a covariate for each of the two neurons and see if there's still synchrony after you have the covariate, just like we do in every other part of statistics. And here the question is, in terms of the model, do we get the log of zeta of more or less equal to 0? Because if we do, then zeta was 1 and we would have independence conditional on the covariates. So again, we model each neuron spike trains individually. We look for the synchrony enhancement. We get inferences by a parametric bootstrap. Just in terms of the magnitude, I thought I'd mention in the data set that I just showed you, we would have 24,000 2x2 tables for each pair of neurons. When we look at 3, which we also did, triples of neurons, we then have, it's 2 to the third, so it's 24,000 times 8, which is 192,000 estimated probabilities. But the thing that makes this tractable is that we actually don't have a large number of parameters. We have just dozens of parameters out of all this. So I said I wanted that these two pairs were really different. They're different with regard to their synchrony. Actually, both of them have synchrony when you start off and just ask about, does the stimulus alone drive these neurons in such a way to make them fire together more often than they would by chance? And the answer is yes. We have strong evidence of excess synchrony. However, when we introduce the covariate for this anesthetic-induced slow wave activity, they have very different behavior. The one on the left, the pair on the left, have synchrony that is due to slow wave in the sense that this log of zeta is approximately 0, 0.06 plus or minus 0.15, whereas the pair on the right, even when you introduce the covariate, you still get a non-zero log zeta. It's 0.82 plus or minus 0.23, which translates into double the number of synchronous spikes, more than double the number of synchronous spikes as you would expect, compared to what you would expect under the null hypothesis of no extra spikes. Now, one thing that's going on here that I didn't mention is I pulled out this pair of neurons just to illustrate nicely the concepts we were talking about. Actually, we had more than 2,000 pairs that we looked at, and so you say, well, what about, don't you have to adjust for multiplicity? What about your false discovery? And well, yes, that's an issue. So the way we're doing this is we're dealing with it in a Bayesian way, or what Brad Efron anyway calls empirical Bayes. Other people call it empirical Bayes. And this is, I think, what he probably talked about last year, empirical Bayes control of false discovery. So the idea is really simple. We have these log zeta hats that we get from that model I was talking about. They have standard errors associated with them, which we got from the parametric bootstrap. And now we say, okay, underneath that, there's some true value, and we want to know whether those true values are 0 or not. So we say, okay, it's a mixture. That is to say, with some there's a point mass at 0, and with some probability they are 0, and with 1 minus that probability they are not 0. So this is the alternative where there would really be an effect, and this is the null case over here. And the point is, we can consider this effect where there really is an effect. This probability density function can be estimated non-parametrically, and that's what we've been doing. I've been doing this with James Scott, who is a former student of Jim Berger's. It's based on stuff he had done in his thesis, and it's very similar in spirit to what Brad Efron has talked about and published about. Work in progress. We're trying to write it up now. I'm just going to show you a couple of results from this. What you get out is something that's actually very similar in practice to a false discovery rate, slightly different, but it's useful for other purposes beyond just saying which are the real effects, and I'll mention that in a second. So we get posterior odds in favor of the alternative, which is what's graphed here, and the ones with big posterior odds are the ones that seem like real effects. And here are the ones where there's enhanced synchrony. This log zeta is bigger than 0, so zeta is bigger than 1. And here are the ones where there's suppressed synchrony. Log zeta is negative, That means zeta is less than 1. We can draw graphs of these using standard visualization software. You can see where there seem to be little networks of suppressed, I think red is, yeah, synchrony suppressed neurons, pairs I should say, and these are, the black is little networks of apparently synchrony enhanced neuron pairs. Here's something that was new and that we're hoping will be of interest to the neurophysiology field, literature, visual perception literature. We looked at the relationship of this posterior probability to the distance between the neurons. So there's an array of electrodes, and that's what's defining the distance. The adjacent electrodes are 400 microns apart. You know the distance between all the different electrodes, and now you say, well, which electrodes did the pair, which electrodes were they on? Measure how far apart they are, and now it turns out from this plot it looks like for excess synchrony, they are closely clustered in space. And there's some theorization about why that would be in terms of the neural networks. Proximal neural networks tend to share things. But interestingly, for the suppressed synchrony case, it appears that there's not this distance relationship. So we get this from a visualization. What we needed to go back and do was to formulate, to get a precise statistical inference, and that can only come from a statistical model. So without going into the details, we enhanced this model to allow for differential effects between the enhanced synchrony case and the suppressed synchrony case, and then we look at the probability of an effect as a function of distance. And it turns out that there is a statistically significant, or however you want to put it, posterior-based effect, where there is a differential effect in terms of distance between these two cases. And that's the neurophysiological finding. The point here is we started with a statistical model, we went to a visualization, we then built a new statistical model from which we could get statistical inference, and from that we get scientific inference. A more dramatic example is diffusion, comes from diffusion imaging. This is Patrick Foley's poster, which he just presented, and I'm just going to repeat a little tiny bit of what he said. Diffusion imaging is an important, relatively new technology which tries to image the white matter in the brain, which are the heavily myelated neuron bundles, which travel over substantial distances, and therefore it gives you a sense of which parts of the brain are connected. This is the kind of visualization you get out of diffusion imaging. Here's another such visualization where the parameters are set up somewhat differently. I think it's the seed, what were called seed values are done differently. But lying behind this, so I'm fond of saying that neuroimaging is not really about making images, and this is often true about visualization. It's partly about making nice things you can look at, but it's fundamentally about solving scientific problems. So here's the kind of scientific problem that would be of interest. It's being applied to, this technology is in the early stages being applied to traumatic brain injury, heavy concussions, serious, strong concussions, to try to identify what's been damaged. And this is a case where they contrast a patient with a normal control, or they also look at within the patient the left side of the brain versus the right side of the brain. And what's graphed over here is a particular measure of the volume of the bundles here in a particular region. And the point is supposed to be that on the good side, the right side, you have a much higher volume than on the bad side where there seems to be damage. So for scientific purposes, people have started to use diffusion imaging across many subjects, and they do things like they'll make some kind of measurement like this, and then they'll do like a t-test. You know, this difference, good and bad, across many subjects, this allows them to say, well, autistic subjects are different than non-autistic in certain respects. That's been an important advance. Or, you know, people with certain kinds of face recognition ability, which came up a little while ago. Some people are very good, but also some people are very bad at it. And, you know, what's different? Well, they can see there seem to be some connectivity issues for people who are very bad at it. But this is using this kind of technology, taking gross measures like these for volume of bundles, and then just looking at that number across many subjects. To my way of thinking, what you really want to do is work within subjects, and especially if you're looking at it as a diagnostic tool, you have to say, is this drop here, is that going to be something that's real or not? Because if you were to do the same imaging methodology on the same patient over again, you're not going to get the same answer. It's not a precise method, and there's tons and tons of things that go into, there's sort of bells and whistles in this technology, and as you change those, you get different visualizations. So, the problem is now to perform statistical inference in the context of the visualization, and I think that's often the case, and that's what Patrick's poster was about. So to finish up and try to draw this all together, I said that there was a big change in the field during the time that I've witnessed it, with the most fundamental points being that modern regression time series methods have become effective, not hard to accommodate non-Poisson variation. This is stuff that I think a lot of neurophysiologists have now recognized and incorporated into their thinking and their methodology that wasn't there before about this time, 1998 or so. I also talked about the synchrony assessment, and I guess I wanted to emphasize that all we did was we adapted and extended statistical models. There was some work involved in a way that I confessed had stumped me for a while, so it's not, in a way, it's not trivial, but it's not new. I would not say it's big new ideas. It's just adapting and extending, and this is, to me, what applied statistics is about. And again, we use standard inferential tools, that have become standard, bootstrap and posterior simulation. So, okay, statistical thinking. One of the things that I found interesting is to go back and look. I like to look at the history somewhat superficially, not as a historian, but just to try to understand the concepts better. I think a lot of times it's good to see how they developed and what the problems were that people were trying to solve. And when I did this in neurophysiology, I realized there's a pretty nice parallel with statistics. They are two fields that are about the same age. So around 1900 and a little after, you have this very basic work that's being done. In statistics, I put Pearson, Fisher, Jeffries here, and there are big names in neurophysiology as well, about the same time, kind of getting the field off the ground. And then we have this heavily theoretical period in here, 1950s. You kind of see a lot of theory in neuroscience as well, some important movement in the field. And then very big developments in this 60s and 70s that kind of are the precursor to what I would call the modern era. One of the things that draws the two fields together is that by the mid-1960s, it was recognized that spike trains can be viewed as point processes. In fact, they're one of the main examples in like Cox's work on point processes. But generalized linear models, software, computational power was not yet there, but was there very soon after. In the 70s, late 70s, early 80s, it was there. It was available. Yet, if you look at this kind of simple stuff and you look at the more sophisticated stuff, the gap is 40 years. It's not just a little gap, it's a bigger gap. The question is why? Why did it take so long for the neurophysiologist to do what we would call kind of the right thing, in terms of statistical analysis? And I would say it was just an absence of statistical thinking. They didn't think statistically. Emery Brown and I, in order to do these review articles, we went back and looked at the literature, and we were struck by the fact that people were often very sophisticated mathematically. There's a lot of physicists in neuroscience. They would do all kinds of clever-looking things, but they weren't acting like statisticians. And we kept asking, okay, so this is bothering us, but what does it mean? What's different? What's different about what they do compared to what we do? And I talked to a lot of people, and I feel like I've been able to articulate what's different in a very simple way. I believe I'm just saying what most statisticians think. When they talk about statistical thinking, we put this in an article called What is Statistics in the American Statistician. I think it comes down to primarily two things. One, we use statistical models. So when I look at what the physicists would do, they do some clever data manipulation, but there's not a statistical model there. And here, the point of the statistical model is to describe variation and turn it into knowledge and uncertainty. There's another reason why and by the way, I don't mean parametric models. I mean, this could be highly non-parametric, but it's always probability describing variation. That's the common thread. And one of the reasons we do this is not only to get things like confidence intervals and to do formal statistical inference, but it's also to analyze the procedures. How well is this method working? And one of the things I found frustrating in reading some of these papers was it sounded like, oh, maybe this is a good idea, it seems intuitive, but they wouldn't show me that it's a good idea. They wouldn't do the simulation study or the math study that would, analyze the procedure. So I think these are the two key things that bring in statistical thinking. And again, in our context, it's just modern regression. This is the source of statistical thinking, of the sort of the workhorse of statistical thinking for us. All right, one last picture in closing. I mentioned earlier that there are many, many different disciplines that contribute to neuroscience, neurophysiology. One of the things that makes it a little bit challenging to get into this field is you kind of have to learn a little bit about a bunch of different disciplines. But I've been at it now a long time, and I feel like I've gotten the jargon and I've gotten the lay of the land, and I kind of know how they all fit together. I feel like I can talk to people, and maybe I don't understand the details of what they're telling me, but I can ask the right questions pretty well. And I thought I'd give you the statistician's view of cognitive neuroscience. How do these all overlap and interact with each other? Here we go. You gotta put statistics in the middle. But one more thing, one more comment. There's a little bit of content here. It's mostly a sight gag, but it's a little bit of content, which is how do neurons carry information? That's what I started this whole presentation with. I hope I've shown you enough to make you agree that though in answering the question, this fundamental question, how neurons carry information, there is no way people can answer that without statistics.